outside and you need to shut the door, but if not, a lot of fun to be here to make this introduction. I know none of you came to see me, and if you're at all familiar with Monterey Bay, you probably already know much about today's speaker, who's kind of a fixture here in the Bay in regards to both his research, but also I think his devotion to educating and helping young scientists find their way, especially underwater, which is the reason why I'm up here. So Jim Watanabe has been in the area for quite a bit of time. Did his PhD on a snail that has changed name. Is it Bactitagula or is it still Chlorosoma? Did a great PhD on Tegula, which were a bunch of fun papers that were a lot of natural history, a lot of observations, but some really cool experiments at a time when we were really just starting to get that stuff done in the subtitle. And a lot of that had a lot of impact on me when I was here at a Moss Landing student. Because when Jim was done, he joined the Kelp Research Program in the division at Monterey Bay Aquarium. And I was their last kelp forest intern way back when, with Mike Foster was also in the audience and a bunch of others. And Jim was my, I don't know, mentor in regards to all things aquarium and that data set, which was a tremendous data set you guys did at that time. And it was great. Got one of my first publications out with all that, and got to do a lot of diving with Jim, putting out pressure transducers to measure waves, which phycologists weren't supposed to be doing back then. And then he moved on just across the alley over to Hopkins Marine Station permanently, where he's a lecturer and teaches a variety of aspects of ecology and natural history, mostly the subtitle class, I think, where a lot of you might know him. Definitely a lot of my students interacting with that way. He basically, and we'll see how this goes, he's the subtitle version of John Pierce, or John Pierce is the intertitle version of Jim Watanabe, either way.
No, I learned from John.
Just encyclopedic knowledge of natural history and patterns, and especially interactions, species interactions. Jim's been really great in not only observing them, but getting others to learn how to do so. In fact, one of the last papers we have of sea urchin barons in the Monterey Peninsula came from a paper you guys wrote way back in the early 90s, and as you guys all know, it's a little bit heightened right now with interest in that. So if you want the historical perspective, and you're going to be working on sea urchins, Sloan, make sure you get Jim aside and hear a little bit more about what the last urchin baron looked like. And then he was the WSN Naturalist of the Year in 2005. Again, I think a testament to the fact that he just really likes to talk to people about what's going on in natural systems. He spends an enormous amount of time out there, and we're just really stoked to have you here, Jim, to give us your presentation. Thank you.
Thanks. Well, thank you all for coming, and thank you for Diana for inviting me here. I was really flattered to get the invitation to come and talk to you, and then had to figure out what I was going to say. And thinking back on, like Mike alluded to, I've been here around the neighborhood for a long time, so I figured I would take that sort of a perspective and sort of in the form of a number of different kinds of lessons that I've learned. Now, as Mike mentioned, I'm a lecturer over at Hopkins Marine Station, and Stanford lecturers we're supposed to teach undergraduate classes, so I'm not allowed to even be a PI on a grant or go look for money to do research or anything like that. So in order to get my data collection fix, I've sort of figured out a good way to do that, which actually has turned out to be quite useful, is enslaving my summer students to help collect data for me. And I like to think of it as a win-win, since they get to learn how to do those kinds of things and the experience of doing it, and then I get the data we can collect. So what I'd like to do today is, over the span of time that I've been playing around in the kelp forests of Monterey Bay, is put that in the framework of a number of different kinds of lessons that the kelp forests have taught me via the data that, with the help of my students over the many years, I've been able to collect. So one of the first lessons is, of course, a rather trivial one that we all know, and any ecologist or naturalist knows, is that things aren't the same everywhere all the time. And as an example of that, this is just some of the data that I've made my kelp forest class collect. These are abundance data in terms of numbers of plants per 10 square meters of adult macrocystis, which we just sort of arbitrarily defined as plants bigger than 4 fronds, because those are the ones that start to have substantial reproductive blades on them. And the red trace is the abundance every summer at Hopkins from 91 to last summer, 2017. And then the blue is from Otter Point, which is just a couple of kilometers away. And you sort of squint your eyes and you see that these things aren't running exactly in parallel to each other. But when you do a scatter plot of them and then do the correlation coefficient on it, it's not very high at all, 0.13, suggesting that that's just a shotgun blast there and not much coordination in the fluctuations of the species, even though these two sites are only a couple kilometers apart. And it's not just the kelp plants, but it's some of the other more obvious abundant, or at least formerly abundant invertebrates, like Pleisaster giganteus, the predatory sea star. It was our dominant one subtidally. Actually, that is significantly different from 0, but note the difference in the scale on either axis here, with the stars being nearly twice as abundant most of the time at Otter Point than they are at Hopkins. And then for the bat star, Pteria, that's also significantly different from 0, but still not tremendously high for such a ubiquitous and abundant sort of species. And so these are just 3 examples of that truism that things aren't the same everywhere all the time. And depending on what scale of either time or space that you look at any of these organisms, you sort of find that same pattern. There's differences. Now, these 3 things, the macrocystis, the giant kelp, these 2 sea stars, these are really abundant things, and we find those guys everywhere. But most of the species that are out there are really rare, and if you tried to sample them with any sort of logistically feasible means, you'd find, most of the time their abundances are down here on the 0 end. And so that's my next lesson that I've learned, is that most species are really rare, and then every once in a while, if you're sort of lucky enough like I've been to be able to sit around and watch one place for a long enough period of time, every now and then those rare species become really abundant. And one of the first ones that happened was the tube snail, Circulorbis squamigerus, which is a gastropod actually, even though it looks like a tube worm. It's got this scaly tube, and it makes its living spinning mucus webs out into the water to capture particulate material. And it was, when I first started diving here in the early 70s as an undergraduate, it was here and there, and I learned to recognize it and see it. But in the late 70s, early 80s, somewhere in there, there was this massive settlement of these guys. And they recruited, especially in the intertidal, down toward the breakwater and at Hopkins. These guys just formed a band of continuous tube snails. And then they were quite abundant in the subtidal, too. And then gradually, that recruitment event sort of declined, although we still find clusters of these guys because their larvae settle gregariously. So that was this first big blast of tube snails, and then they sort of declined. Another one, and we'll come back and talk about this guy later on in the talk at several points, is the acorn barnacle, subtidal cousin to Balanus glandula, the intertidal barnacle. This guy was always, from when I first started diving here in the early 70s, kind of rare and patchy. You'd find it in sort of disturbed places like old holdfast scars of giant kelp plants, and other places where space opens up temporarily due to small-scale disturbances. You'd find these guys recruiting in there. And then, for several years, during 3 pulses, early 2000s, 2001, 2, and 3, and then 2010 to 2014, they were everywhere. They were just settling on everything. It was wall-to-wall barnacles, sort of like this close-up picture here, but everywhere. And even some of the soft-bodied things, like the big gumboot chitons or the big giant keyhole limpets with soft parts showing, even those guys were getting covered with these barnacles. So they'd settle, and they grew fairly quickly, and like all barnacles that that settle in very dense aggregations, they grew up in these big, tall, columnar shapes as they got really pressed for space and then peeled off very easily. And so they covered a lot of space, but from the point of view, if you were a little larva looking for a place to settle, from a larva's eye view close to the bottom here, these are all the legs, the feeding thoracic appendages of these barnacles. And so it was like, you know, barnacle hell out there. If you were a larva, these things would just probably eat you before you could get to the substratum. And it was like this for several years in the early 2000s and again in between 2010 and 2014. It was just barnacles everywhere. And we'll come back and talk about this a little bit later because it has some other interesting aspects to its story. Now, it didn't sample the barnacles, just noticed that they were really abundant. But along the way, I was having my summer class again, collect other data on feeding and feeding frequency and the prey items of Pisaster giganteus, the most abundant predatory sea star in the kelp beds. And this is the frequency of Balanus crenatus in Pisaster diets. So this is the fraction of feeding stars that were feeding on Balanus crenatus in each of these years, and I'll let you guess which years Balanus was really abundant. Because Pisaster, again, as we'll see a little bit later on, we'll see a little more detail of its diet, but it has an extremely broad diet, and it's very opportunistic. So when there's a lot of easy-to-get prey around, it will concentrate on those. And it was during these years of really strong barnacle presence in the kelp beds, both at Hopkins and at Otter Point, that we saw really high frequencies of barnacles in the diets. So in lieu of having my own data about barnacle abundances, I just sort of asked the starfish, what were you guys choosing and eating, and managed to get some information about the probable abundance of barnacles during these years. Certainly, these are the years when the barnacles were there. Then the third species that has unusual occurrences in terms of its abundance are the sea urchins, both Strongylocentrotus purpuratus and Mesocentrotus franciscanus now. And these are data, classic data from John Pierce and Tuck Hines' paper in Marine Ecology Progress Series back in '87, where in the Hopkins kelp bed they documented this really surprisingly strong recruitment of sea urchins, which didn't last for very long and then declined relatively quickly within, say, a couple of years. And then after that, sea urchins in the Hopkins kelp bed stayed pretty much flat down here at less than half an urchin per square meter. And note the discontinuities in the time scale here. This is 40 years long here. So these were John Pierce's data. These are Theo McKelly's data from when she first arrived at Hopkins. And then when she stopped doing that, she handed it off to me in the summer class, and we did it for a couple of years, and there were just so few urchins around at that time, we just stopped counting them because there were other things we wanted to do during the class. And then, of course, they've recently gone way up in abundance. But with this 35, 40-year gap in between those big peak events. And we'll come back and talk about this a little bit later, some of the causes possibly behind that later on in the talk. But I think these are three good examples of the fact that most species are rare, except for every now and then when they show up and can be surprisingly extremely abundant. Now, the third lesson that the kelp force has taught me is that the historical context in which even extreme events happen are important, and that we have to pay attention to those things. And the more we know about that current ecological context when some of these big unusual events happen, well, I don't know if we can understand them better, but at least we have a better idea of what's going on. So to develop this story, it's going to take a few slides here. But it started out with initiation of these permanent photoquadrats. We started. So in 2011, again, by enslaving my summer class to drill holes in the granite out around the Hopkins kelp bed here, we put in a pair of eye bolts marking these photoquadrats. So there's 52 of them. They're a half square meter in diameter, and we set them up mostly on more vertically oriented rocks, simply because those places have less three-dimensional cover of many layers of algae and so forth. So figured it would be a little bit easier to monitor the sorts of things that are going on in these plots, at least photographically, if they were on vertical plots. And we divided those across 3 locations. So the Hopkins Marine Life Refuge West, Central, which is straight out off our big bird rocks area, and then the east site is right around that same study site where John Pierce did all his work, and a lot of other folks have done their research work, right in the central portion of the east side of the kelp bed. And then I've been going out every 4 months, in March, August, and December, since August of 2011, and shooting photographs of these plots. So March is right at the beginning of the upwelling season and right before most of the recruitment windows for a lot of the different species starts. August is sort of the tail end of the upwelling season, and after a lot of the recruitment has gone on. And then December is usually right before we get our big, huge winter storms coming through. So I figured those were good times of the year to take these images. So what we're going to do next is take a look at a series of images from one of the quadrats through part of the time series, just get an idea of what these things show us. So this is a picture of one quadrat in August 2011. All of this white stuff here, actually the quality of the real photos are better than this. You can actually zoom in and see what these things are. So these photographs here are a little bit reduced in resolution. But this is August 2011, the first batch of photographs for this particular quadrat. And all this white stuff here and over here is Balanus crenatus. So remember, the Balanus crenatus started their big, most recent settlement event in 2010. And so this was in the second year of that big settlement event. So this was August. And then by December, there are some patches missing in the cover of the barnacles. Still fairly heavy barnacle cover on this side, but thinning out a little bit, you can almost see some patches of crustose corallins in this corner here. And then by March, barnacles are almost all gone. So from August to March, most of the barnacles disappeared. There's still some right in here, but there's corallins showing here, some erect foliose red algae, rhodymenia, and a few other things showing through here, with a much reduced cover of barnacles. And this is March. August, barnacles are back. So between March and August, there is another huge settlement of Balanus crenatus covering over all the stuff that was here, except for these few hardy little sprigs of red algae here, but covering, again, most of the quadrat. Then by December, thinned out again, with even corallins showing through. March, almost all the barnacles are gone. Some patches of encrusting bryozoans here. Predatory barnacle predator snail we'll talk about in just a minute. And then again, August 2013, barnacles are back. So what I've done here is I went and just sort of did a quick and dirty eyeball of at least 10 quadrats in each of the 3 locations here and qualitatively scored them for barnacle abundance from 0 to 5, with 0 being less than 5% cover and 5 being greater than 85% cover. And then I've plotted the median barnacle abundance amongst those 10 replicates in each of the 3 sites on this. And you can see that the 3 sites were doing the same thing. So again, this event started in 2010 before we started the photographs here. But here we see high numbers, high cover of barnacle in all 3 sites in late in the year. This was August and then December. Lowest in March, way up at the end of the summer, drops again to its lowest values in early in the year, in March, peaks in each of these years in late summer. And then, interestingly, after 2014, barnacle settlement pretty much stopped. They just stopped coming. And in most of the quadrats in most of the sites, there's hardly any barnacle cover for the last 3 years. And by 2017, December 2017, this is what that same quadrat looks like. So over on this side, that used to be just completely covered with barnacles. We have probably 5 or 6 different species of erect bryozoans, some of the crustose algae, foliose algae showing, big compound tunicates here, so a much richer benthic cover in the absence of those barnacles. And even these white things here, which in this resolution of the slide looks like maybe barnacles, what those are serpulid polychaetes. It's a little calcareous tube polychaete. So, much different look to the community after the cessation of these barnacle settlements. So, of course, this up and down, you might ask, well, what's causing that? And those of you who are familiar with these sorts of vertical rock wall communities probably already has a good idea that the driver on that was predatory sea star Pisaster. So this is Pisaster giganteus. During the time, especially when there was lots of barnacles here, I take a photograph of the plot as it sits when I first get to it, and then I peel off all the stars and all the other big things that are in the quadrat, and then take a picture of the stuff that was underneath all those guys. So this is one of the ones prior to peeling off all the sea stars, but that's not a really unusual picture of these, particularly the west side quadrats during the times where there was all these barnacles there. There were just tremendous numbers of Pisaster eating these barnacles. All right, so driving, probably one of the 2 things that were involved in this cycling here was, first of all, really strong annual recruitment during these years of the barnacles themselves, and then the predatory consumption of those barnacles by Pisaster. And then everybody remembers what happened here. 2013 was when we had the big sea star die-off. And in the Hopkins kelp bed, we lost maybe 75-80% of the Pisaster by number in terms of their densities. And so real decimation of the predator population, but then what the heck's going on here? Because we saw that reduction to a very low level of the barnacles during their last year of big settlement, rose to a peak, and then declined yet again with only 20% or so of the Pisaster there that were there before. And so what's going on with that? And one of the things that was happening at the same time was changes in abundance of another one of the predators. And there's this big snail that lives out there too, called Ceratostoma. The leafy hornmouth is one of its common names. But it's a barnacle predator. It's a barnacle specialist. And drills and eats a variety of other invertebrates, but when barnacles are around, it will choose those and eat them voraciously. These guys lay benthic egg cases, and for the purposes of our story here, one of the most important characteristics of their life history is that they hatch out of those egg cases as crawling juveniles rather than swimming planktonic larvae that go off who knows how far away before they settle. These guys crawl out of their egg cases as little baby snails. And so it allows them, when there is a bonanza of potential food, like the barnacle settlement years were, allows them to take advantage of that and locally increase their population fairly quickly. And so this is one of the quadrat corners, and you might say, oh, what's in that picture? And if you look really closely, these are Ceratostoma snails covered with corallins and so forth. And if you count all of them, there's about 30 or 32 of them in this corner of the quadrat, getting together, mating, and getting ready to lay egg cases. And so this is an indication of how abundant these guys, who had, prior to the settlement of the barnacles, have been relatively rare. Then they would lay egg cases. This is a corner of another quadrat where almost a quarter of it is covered by egg cases of Ceratostoma. And it was during this time that this is the only time in the last 40 years that I've been diving around here that I saw little baby ceratostoma. These guys were about a centimeter long. Actually really cute if you think snails are cute. So, and again, this is another one of the things that we weren't counting. So I didn't have numbers on them. So the next best thing is to go back to the photographs, the plots, and look at those. And half square meter is probably not the optimal size to sample a relatively small snail like this, say compared to the sea stars. But you can actually see, even in these counts, which are just the totals for each of the locations, totals of all the plots, the numbers of ceratostoma that are visible in them. And you can see that they're getting more abundant. They really become obvious after the Pisaster mortality. And I think part of that is, in addition to both Pisaster and ceratostoma eating the barnacle, Pisaster also eats the snail. And so when the Pisaster are really abundant on these plot locations, the snails are probably avoiding those because these guys have an escape response to Pisaster. But at least you can, if you use your imagination, you can see some response of the population of ceratostoma, particularly in the west side quadrats, to this fluctuating but consistent abundance of prey for these guys during the multiple years that went on prior to the disappearance of the sea stars. And because there was sufficient food available for a long enough time, and that these guys have benthic reproduction with crawlaway larvae, or crawlaway young, they were able, their population grew enough so that when the Pisaster got decimated by whatever it was that killed them, the populations of the ceratostoma had risen high enough to almost take over that role, causing that annual decline in the barnacle abundance, and then when the barnacles settled again, causing it to decline. And then when the barnacles stopped settling, I think these guys have had a harder time finding food. So finally, this is where the point of all of this story comes out, is that the history and the context of when these events happen makes a difference. Because if you think, what would this set of plots look like if the mass mortality of Pisaster occurred at other times? So for instance, if the Pisaster had died off prior to the first year of the big barnacle settlement, there would have been probably qualitatively number 5 covers of barnacles, and they would have persisted instead of having this annual fluctuation, because the Pisaster wouldn't have been there to drive their populations back down. If the Pisaster had disappeared here, the barnacles wouldn't have been around for long enough for the ceratostoma to respond and increase their population size to sort of take over that role of causing these fluctuations in the barnacle population, at least for several years. So the populations might have looked a whole lot different, and also we might have inferred that because the Pisaster disappeared here and the barnacles showed up the year after, that prior to that, the Pisaster were controlling the barnacle populations and not allowing them to take over like this, and once the Pisaster disappeared, that's why the barnacles showed up. So we would have been lulled into that interpretation if that event had happened here instead of the time when it did. If the Pisaster had disappeared after the barnacle settlement years had happened out here, we probably would have had to try and scratch our heads and figure out what the relationship between Pisaster and the increasing diversity of the quadrats that we were seeing out here after the barnacles stopped settling. And we would have missed the importance of the role that potential replacement role that ceratostoma could play. And then lastly, if the mass mortality had occurred where it did and we hadn't been paying attention to what else was going on in the habitat around these events, and keeping track of the fact that ceratostoma populations had been able to respond to the high abundance of prey in the previous years, we might have discounted the strength of the role that Pisaster was playing in these fluctuations, because we saw that same fluctuation occur again after the Pisaster had disappeared. And so when big events happen, the other events that are happening around it are very important, not only for the actual ecology of the communities that are out there, but for our interpretation of what the involvement and importance of those different components in the community are to the responses to those rather large changes. So what this tells me in my list of lessons is it's really important to not just go out and look at one thing, but to look at the community that's going on as a whole, and that sometimes some of these rare species may be playing important roles that we might not notice if we don't pay attention to them. Okay, next lesson. The simple abundance of a species, I've found out, doesn't necessarily tell you what it's doing out there, okay? Because we ecologists, we love to count things. We love to go out, count things, and put them down in our little spreadsheets and keep track of numbers. And a lot of the really great programs that involve citizen scientists to go out, like Reef Check, to go out and sample things, and the PISCO program that's continuing to count things out in the environment. And that certainly tells us what how those population sizes are fluctuating, but it doesn't always necessarily tell us what role those species are actually playing in the communities. And at least to me, that's an equally important component. So we'll go back to Pisaster here, because we have data from my class again on Pisaster. So this is the abundance of Pisaster that we counted in my class every summer since the 90s here. And what you see is, if you kind of squint your eyes, you see that the long-term average here is probably just below two stars per 10 square meters, with really large standard errors each year, fluctuations up and down, because these guys also occur very patchily. So some years, some of the quadrats happen to get into clumps that look like this, and we get these big numbers, but also big standard errors on them. So some of this fluctuation is probably sampling error, and in general, it's probably been about 2 stars per 10 square meters up until the mortality event here in 2013, when, again, about 80% in the Hopkins kelp bed of these guys disappeared. And much smaller standard error bars suggest that these guys are uniformly not very abundant everywhere we sample them. And having known this star for a long time and seen the things that it does, and one of the additional things we'll see on the next slide is the diets of these guys, which is yet another thing I've made my summer class do. I make them learn all the species, and then we use that when we go out. We sample the stars and quadrats, turn them over, and see who's eating, and if they have their stomach out or they're holding on to prey next to their mouth, we try and identify what it is they're actually eating. And so we've been studying these guys, looking at their abundance, seeing what they're eating, and when this mortality event happened, I thought, oh, we're going to see some really big changes. There's going to be some big effects, because how can the most abundant benthic predator out there in the kelp bed get reduced by 80% and there not be any obvious effect? But there wasn't. At least from what I saw, there was no real obvious immediate change in the rest of the benthic community to the loss of Pisaster. I scratch my head. Why can that be? There's got to be a big effect that these guys are having. And I went back and I was looking at the diet data that we've collected. And this is one of those slides that you always tell your students, never make a slide like this. Never make a slide that's got all that stuff on it for people to read. But actually, the amount of stuff that's on here is the whole point of the slide. So these are our diet data from, for Pisaster at least, from both Hopkins and Otter Point, a couple kilometers up the coast beyond Lover's Point from Hopkins. And so, like I said, every year we go out and sample the diets, drop randomly placed quadrats, look at the number of stars, and then turn them over and see what they're eating. And so these first numbers here after the date, these are the numbers of stars that were observed that particular year, and then the proportion right next to it is the fraction of those stars that we're actually feeding. Okay. And then listed are the top 2 prey items of the stars that we're feeding, and then the fraction of the feeding stars that were eating that particular prey item. And so they eat a bunch of different things. Serpulorbus and Petaloconchus are vermeted gastropods, so these tube snails. Tegula, Pomelax, Ceratostoma, Calliostoma down here, those guys are all good card-carrying mobile gastropods. Here's our friend Balanus cornatus, the lilleacorn barnacle. Again, huge fractions of the feeding snails or feeding stars, and higher feeding frequencies in these years where there was a lot of Balanus around, because they're really easy and really to eat and really abundant. So the stars were really concentrating on those. But other than these years, there was a lot of really easily available, abundant, superabundant prey. So 2001 to 3, and then 10 to 14, when the stars were eating the acorn barnacles. The other years, for most of the years, adjacent years, the top prey items are different, and within the same year between the two sites, the top prey items are different too. In total, we've documented somewhere around 80 prey species that Pisaster is eating in either habitat. And now, about 30 or so, about a third of those have been observed only once. So some of those you probably have to be a little suspicious of the identification of the prey. But still, even if we set those aside, there's a diet that contains about 50 species, and they're not exactly the same 50 species in either of these two sites, suggesting that Pisaster has a really broad diet, and it's a really good opportunist. And so whenever things are abundant, that are easily catchable, that's what it's going to eat. And in years when those things aren't that abundant, and there are less catchable things around, then their diet can vary from place to place and time to time a whole lot. And so being such a good opportunist with such a wide diet, I think that's the reason why we didn't see such a huge effect of the loss of the Pisaster. Because it has that broad diet, it's very opportunistic in the prey that it chooses, and therefore the effect on any one single prey species is much harder to predict, okay? Because you don't know for any given year which prey might be the most abundant or most easily catchable, and you don't know what they're going to be eating. So because it varies from year to year, and it's mediated by the quasi-independent dynamics of each of those other prey species, the effect of removing the Pisaster may not be all that predictable. Now, like we saw in the Balanus, under certain circumstances, for things like Balanus that can't run away and they're kind of stuck in one place, and the Pisaster can eat them very, very easily. Under certain circumstances, Pisaster can indeed affect the abundance of its prey because it caused that fluctuation that we saw in the photoquadrats. But on a lot of the other things, the effects of Pisaster was probably a lot more diffuse and very indistinct because of the wide array of choices that individual stars can make for their diet, and the very opportunistic nature by which they choose those prey. And so just by knowing how many stars there were, because of the nature of their diet and the potential effects and varying effects on a wide array of species that are fluctuating independently of one another through time, it's really hard to predict what the exact effects of those guys are going to be. So even though we know that Pisaster decreased in abundance by 75 or 80%, it's really hard. You can't really say that its effect on any one of its prey species changed by that same amount, because it changes from year to year what prey they're eating and which things are available to them. Now the other one where this lesson sort of has been brought home has to do with sea urchins. And again, this is a plot of abundance of the urchins. This is John's data from the Hopkins site, and then these are Theo McKelly's data, and then these are my class's data here. And unfortunately, we have this gap here from 2013, 2012, 13, and 14. And of course, the sea star mortality occurred right there in the middle of it. And so you look at this side of the sea star mortality, and you look at that side of the sea star mortality, and like a number of people have said, That's why the urchins went up, is because the sea stars disappeared. And there's a number of reasons why I think that there's probably not a cause-effect relationship there. And for the following reasons. First off, here in early 2014 was the first time in the photographic quadrats that I saw a sea urchin show up in those quadrats. Remember, those are vertically oriented quadrats, and so they're not the first choice microhabitat for a sea urchin. They like crevices and places where they can find shelter, and the only reason that you might expect to start seeing them in these more vertically oriented faces is that there's a lot more other urchins around, and all those preferred microhabitats are already full. So those guys are walking up the walls and the vertical faces. Furthermore, the size of the animals that first show up in the quadrats are about 20 to 30 millimeters test diameter, which suggests they're already at least 2 to 3 years old, which would indicate they settled back here somewhere. So that's one piece of evidence. Second piece of evidence was in January of 2014. Giacomo Bernardi, UC Santa Cruz, sent John Pierce an email about a dive he and Nicole Crane had made out at Point Pinos along the Pacific Grove shoreline. And Giacomo said, "We were diving out there, there was no kelp, and there was all these urchins, and I didn't know purple urchins got that big." And they must have been 60, 80 millimeters test diameter, which again suggests they were probably, optimistically, at least 5, 6 years old, which would have put their settlement back in here. And then the last piece of circumstantial evidence is I was digging back through my dive logs, and in the summer of 2011, we were diving in North Carmel Bay at the Pinnacles offshore reef site. And in my notes, in my dive log, there were all these little places where there were these small urchins, maybe 25, 30 millimeters, in clusters, forming these localized grazing halos around shelter sites. You know, small ones. So these were small urchins, and they were in clusters. They were coming out making localized grazing marks, suggesting again that those guys were small, young ones. And in my field notes from 2010, I didn't notice anything, or at least I didn't note anything like that down at that time. But I did see it in 2011. So for all of these sort of circumstantial evidence kinds of things, suggests to me the urchins were there well before the sea stars died. Okay. And so they may have gotten a little more active. They may have been grazing on things a little bit more in the absence of such a large number of sea stars, but probably there's no causal relationship between the loss of the sea stars and the climb in the urchin population. Oh, last piece of circumstantial evidence. This picture was taken in the Hopkins kelp bed of a little crevice. And again, these guys are probably 25, 30 millimeters across. This was 2008 in the Hopkins kelp bed. All right, so this is a size frequency distribution of just Purpuratus in 2015, 2016, 2017 at Hopkins. And then these are the abundances of both Purpuratus and franciscanus. This is the Point Pinos sites here. You can see abundances are much higher out at Point Pinos than they are at Hopkins, but the size ranges are about the same. And the maximum ones that we're seeing and measuring in these years are pretty big sea urchins in both locations. And so these guys have, especially out toward Point Pinos, like Giacomo reported to John Pierce in 2015, were causing a deforestation. So this is a Google Earth picture of the Pacific Grove shoreline. Here's Hopkins. The aquarium is right here. Lover's Point, Otter Point, Point Pinos. So the deforestation and complete loss of the canopy, pretty much wall-to-wall sea urchins, at least as of last summer, and no kelp canopy except for small clusters in very near-shore parts of the former kelp beds, now occur from at least Point Pinos. I haven't checked this area out here beyond Point Pinos, but certainly from Point Pinos to Hopkins, with this sort of sequential loss of the canopy up to summer 2017, where about half of the kelp bed off Hopkins is now has no surface canopy on it right now, and is got high abundances of sea urchins. All right, so if it wasn't the sea stars, I guess, you know, the big elephant in the room is, or the big question in the room is, what the sea otter's been doing. Because here we are, this is the middle of sea otter country, and everybody probably has heard the sea otter, sea urchin, kelp explanation, and that urchins eat the kelp and deforest it unless the heroic sea otters are there to eat them and control their populations, and thereby have a positive effect on the giant kelp. Everybody knows that story, because it's one of those 15-second soundbite elevator explanations about how the kelp forests work. And the sea otters are the guardians of the kelp forest because they eat the sea urchins, right? Okay, so what have these guys been doing? Okay, because here we are right in the middle of sea otter country. In the last few years, otter abundances have been fluctuating, but in the southern Monterey Bay area, they've probably actually gone up just a little bit. And so it's not like we've lost sea urchins in southern Monterey, or sea otters in southern Monterey Bay. So what's going on? Why aren't they eating all these sea urchins that are out there? And the urchins are just out walking around, exposed. They're not down in crevices. There's no kelp. There's hardly any algae other than crustose corallines. So there's no place for them to hide. So why aren't the otters eating them? And so this brought to mind, again, a lot of these lessons of things that you already know, but we've just sort of forgotten in this generality that we just kind of assume is true. And one of the important things is that otters don't eat small urchins. Okay? So newly recruited small urchins, otters don't even notice them, and they don't eat them until they get probably well above 30 millimeters diameter. In all the years that we've been sampling the diets of piesaster, we've never actually caught a piesaster in the act of digesting a sea urchin. Okay? Never seen one. You see lots of empty tests around, but we've never caught a piesaster actually digesting an urchin. And the big 20-arm sea star Pycnopodia, which does eat urchins, swallows them whole, but around the Pacific Grove shoreline from Hopkins to Point Pinos, Pycnopodia, even though it's been present up until the mortality event, it never was really astoundingly abundant like it has been described in other portions of the Pacific coast. And so it's never been abundant enough to actually probably control urchin populations, at least at the Hopkins kelp bed or along the PG shorelines. And probably, or one of the big possibilities, is that the absence of urchins over that 40-year span from the big recruitment event that John and his students documented back in the 1970s up until 2010 or so probably has been due to a limitation of recruitment and supply of young urchins rather than constant predation, and that the otters have probably been just knocking off the occasional big, dumb urchin that comes out of its crevice and walks around and then gets eaten, because the otters do eat the sea urchins there. But because of really sporadic recruitment of the urchins, that may have been as big a factor in the absence of deforestation events along the Pacific Grove shoreline as predation on adult urchins by the otters. Now, otters, as everybody knows from the work that Marianne Reedman and Michelle Stedler at the Monterey Bay Aquarium working with Jim Estes up at UC Santa Cruz, everybody knows otters eat a very wide diet as a species, but individuals sort of specialize on a small subset of that diet. And when the females have pups, the pups learn how to be an otter from their mothers, and the mothers pass those preferences on to some extent to the pups. And so my thought here, or question, is did persistent low numbers of urchins for those 30 or 35 years there cause a cultural memory loss in the fact that you can actually eat urchins? And because there weren't enough urchins around for any of the adult otters to really specialize on and concentrate on, it didn't get passed on to the kids. And so the kids forgot, and when this huge number of urchins show up again, everybody says, what's those spiky things down there on the bottom? And until somebody tries it and starts eating them, there could be this real time lag. And so you think, oh yeah, nice story, everything. But I think there's a precedent for it. And again, this is one of these things that have happened over a long period of time that you sort of benefit from by being able to sit in one place as sedentary as I've been for the last 4 decades. And this precedent is Colletii, big Kellett's whelk. It's a big predatory gastropod, and it started showing up in substantial numbers in Pacific Grove in the late 1970s. And actually Tim Herlinger, who was when he was his master's student here at Moss Landing, published a paper on the range extension of Colletii up into Monterey Bay at that time. And between the time it first showed up in late 70s through the early 90s, it just kept getting more and more abundant, up to a point where it was about 0.1 to 0.3 per square meters. So you could go out there, close your eyes, stick your hand down on the rock, and there would be a big Colletii within about a meter of your hand. And I always wondered, why aren't the otters eating these things? Because they're big, beefy, meaty snails, and they don't have a shell that's any tougher than an abalone shell or any of the other gastropods that otters eat. But they just weren't eating them. And then, in a really short time, it felt like about 6 months, but it might have been a little longer than that, between 95 and 96, the population of the Colletii had dropped by an order of magnitude, and there were nothing but busted shells all over the bottom. And this was at the time when Steve Lonhart was doing his PhD work. And Derek Stevenson was one of my spring class students in the early 90s, and he worked on these guys. And both of these studies documented these kinds of numbers. And then Steve, in his dissertation, documented the shell breakage and the sizes of the shells that were broken and so forth. But and we talked a lot about this, but he thought that the discrepancy in numbers that he was getting in 1996 was sampling error relative to these really high numbers that had been counted a few years earlier. And I kept telling him, no, it's because the otters have been eating them, and they hadn't been eating them before. And so it took an Einstein otter to go down and sample one of these guys, or a real foodie otter to go down and sample one of these guys and say, oh, these are really good, and then start eating them. And then otters are so smart, they watch what the other guys are doing, and they say, what's Fred eating over there? You know, and, oh, you can eat those. And so they started eating them, and then the population dropped like a rock. But it took 15 years or so for them to figure that out. And so this is the basis of my sort of thought about the otters have forgotten that they can eat urchins, at least in our neighborhood. And it's going to take them a little while to figure it out. But then once they do, they'll clean them out probably really pretty quick. But in the meantime, the urchins are taking out the kelp, and we have these widespread barren areas along the Pacific Grove shoreline. All right, so what this tells me is that life is a lot more complex than we would like it to be. And we know that because we go out and we see it every time we visit these sorts of habitats, but it's really easy to forget when we have a 15-second little explanation of how the system really works. And so, based on everything we know, these things are complex events, and they're complex interactions of a bunch of different things, from variation in sea urchin recruitment and their early survival, to the variety of triggers that alter the feeding behavior of urchins, availability of crevices, removal of kelp plants during winter storms that cut down the drift algal production that they normally feed on when they're sitting in sedentary. All of those things that can change urchin feeding behavior, which changes the effects they have on the rest of the community, even though it doesn't affect their absolute abundance at that particular time. And then, of course, control of those grazers by the predators. And to sort of paraphrase a nice phrase that Mike Foster and Dave Scheel used a long time ago, there's a lot of bricks in this wall. And there's not just a single keystone that makes the whole thing work or holds the whole thing up. There's a lot of interacting moving parts. And it's not really just a bunch of bricks in a wall. It's a bunch of moving gears and parts that are continually changing. And these effects are all nonlinear, complicated, and extremely dependent on the context and the history in which they occur. So the sea otter, sea urchin, kelp explanation isn't wrong. It's just incomplete. And if we want to really understand these kinds of communities, we have to pay attention to the details, even within the framework of these generalities that are not wrong, but just incomplete. So the lesson here, simple abundance of species, either Pisaster or the presence of the otters, doesn't necessarily tell us what they're doing out there. And we have to pay attention to their natural history and not treat species like these little black boxes that always do the same thing in exactly the same way, every place they occur, every time we see them. Because biology is a lot more complicated than that, and it makes studying ecology that much more complicated, but also that much more interesting. Okay, so I've been yammering on for a long time here. I put this slide in just to remind myself that it ain't just about barnacles and sea stars and sea urchins. Every one of the little mug shots up here of these species has a little story. Some of them that I don't have as much data on, but stories of change that have occurred over the last 40 years that have caused me to sort of pause and scratch my head and wonder why that's occurred. So it's not, even though these are probably, in terms of their evidence, the most obvious things that have been going on, there's lots of other these kinds of stories that are out there, and we just have to be in touch with the natural history of these habitats and the organisms that are out there to follow what these stories are. Okay, so last lesson. You can't ever have enough data. You just can't. There's always something else that you forgot to measure. And one of the reasons that we can't ever have enough data is that we can't count what's not there yet. And so these unusual events where things happen, where we get this big spike in abundance of different species, we can't predict those. And so we can't be monitoring every one of those species ahead of time to be able to track their trajectory as they increase in these unusual recruitment events, because we don't know what's going to happen. The other thing that I've come to realize is that we can't really replicate things in time because things are always changing, okay? And so because the ecological context and the circumstances where some of these events occur, they're never going to occur in exactly those same circumstances again. And the responses of the rest of the community that we see to those kinds of unusual events may be very different one time to the next when they occur because of the other circumstances that occur there. And so we can't think of things repeating in time because they really don't. And then a lot of these so-called natural experiments, of course, to the experimental hardcore ecologist types amongst us, these ain't real experiments anyway because there's no controls. And if we know already ahead of time that these communities fluctuate so much through time that we really don't know sort of what the control situation might look like. So even though we know these kinds of things go on and we know that they're extremely interesting, we just there's no way we can ever collect enough data. And so the other reason is this. And so this is, about 40 ticks of the year, the annual clock here. And these are my imaginary potential trajectories of a bunch of different species. And my thoughts on this have been prompted by discussions with John Pierce and Chuck Baxter. We were talking about, these things have changed in the last decades. And we're trying to figure out reasons of why that would happen and what the actual changes have been. And that's been a prompt to a really neat thing that Theo and John mostly have been, Theo McKelly and John Pierce have been spearheading this historical ecology project, where they're getting in touch with all these old timers and interviewing them about what these places looked like back at the times when they were here, to get this idea of how have things changed. You're trying to answer this question of how have things changed. And this has prompted my thinking on a lot of this with regard to this time span here, because subconsciously, the first contact we have with any ecological system, you just can't help but think that that's the good old days. Okay, that's the way it is. That's the way it should be, because it was pristine back when it was pristine to me, so it's got to have been universally pristine back at that time. And then anything that differs from that is a change. And whether we interpret that as a good change or a bad change probably depends on what's changed and the degree to which they've changed. But if in reality the fluctuations of a bunch of different species look like this, something like this, with some things declining over long periods of time, other things spiking, some things maybe going up and then going down again, and then maybe like Pisaster coming here and falling off a cliff and staying at low levels for a while. Depending on where our window is in this progression through time, it will give us a real different view of what those communities are looking like, and what we feel subconsciously, even though we try to resist it, what they should look like. And against this background of a long-term change here, we may have an accurate, or we may have maybe not so accurate a picture of what that system really looks like. And it's only by sitting on these places for long periods of time and being able to look at them that we can at least get some sort of perspective on what that looks like. Because because of the very nature of the way science works these days, either for the duration of a thesis or a 2-year or 3-year NSF grant, we look at things intensely in small chunks of time. And ecology doesn't necessarily always work on small chunks of time, and it takes long periods of time to get this sort of perspective. Okay, so last lessons. And this is if I were able to go back and speak to my former self when I was just a young, snotty-nosed, know-it-all undergraduate that first showed up in Monterey Bay to try and flop around in the water and spend some time looking at these organisms. And if I were able to sort of communicate the lessons that have sort of entered my brain through osmosis over the last decades, it would be this. One is watch places for a long time, which I've fortunately been able to do. Keep better notes, which I probably could have done much better and wish I had. Because there's no substitute for writing down what you see today, which seems maybe a little bit insignificant, but 20 years from now, it could be really important to have that little note with a date on it. Get students to help while they learn things. And fortunately, this is one of the things I've been able to do, and I think it's a great model for doing these long-term sort of finger-on-the-pulse kinds of studies of changes in any ecological community. Certainly, we have to do manipulative experiments when we can, but we shouldn't expect those experiments to replicate in time, because the circumstances are going to be different. And even though there's no percentage in ever repeating an experiment, I think it'd be really good things to do, because by doing that, we can see the variability in the responses of these communities and how the differences in circumstances under which we do those experiments make a big difference. I think we should worry less about p less than 0.05. So we all do our generalized linear mixed model analyses, or we do our big meta-analyses with Bayesian-generated maximum likelihoods and so forth. And we all love to do those things because playing with numbers like that is sort of fun. But we really get fixated on our statistics. And I think we can learn a whole lot more about how these communities work by maybe paying a little bit more attention to the biology and natural history and putting more value on that than p less than 0.05 in the papers that we publish. I think this is a big one. Unexpected events aren't just noise. They're not something that we need to sort of try and ignore or average out. They're part of the fabric of this big tapestry of how these communities work. And somehow we need to change the way we think about how we ask questions of these communities to incorporate that. And I keep racking my brains thinking, you know, if I knew a little bit more, worked a lot harder, or were a lot smarter, I could figure that out and ask the question in a way that would make how we get the answer to it perfectly obvious. And I just haven't been able to do that, so that's sort of up to you guys, especially the young ones in the audience, to think about these questions and think about these approaches and try and take that sort of more holistic view of how all these places work. Not just the things that we can do because that's what we can do in a short period of time and get a paper out of it, but how we can understand these communities better. And I really think it's how we ask that question, and I still haven't quite figured that out. And then lastly, don't think in short, straight lines. Our simple stories aren't necessarily wrong, but they're incomplete, and don't fall into that trap. Okay, so I'd like to just then finish with some acknowledgments. Of course, all the students in my Kelp Forest Ecology class from 1975 through 2017, faculty and staff at Hopkins, and then in particular these people that have either helped me a whole lot or provided inspiration and mentorship, all my stalwart dive buddies across the decades, and then everybody in this room and in other places who've taught me something about the tiny little creatures that live in Monterey Bay. Thank you for that. So I'd be happy to answer questions.
Hey Jim, that was a great talk, and it is really easy to see why you're such an acknowledged lecturer and good teacher of students. Really excellent talk. A couple things I just wanted to make a comment about. Mike Foster and I were at a meeting once, and it was a review of a very large project, and somebody got up and said, every quadrat tells a story. And we thought, oh my God, there's about 5,000 here. This is going to be a very long day. But one of the really interesting points, and I was thinking about it before you brought it up about lessons to yourself, is how modern ecology sort of factors out variance, and often it is called the noise of the system. So while you're, the sort of idea that you're looking for a long-term signal and all these sort of things are just happening that are somehow unimportant in the overall context of it. Well, one of the things I was interested in is you have a lot of things with abundances. One of the things that'd be interesting is to see, just from the way you piece this together, whether a functional aspect, or when is predation important, and to what extent, and when is recruitment important, and how that balances out over a time series, and thinking in not just abundances, but function, and how that might change, because a lot of that stuff you're talking about seems to be recruit and behavioral sort of dictated, and it's a really common thing against a lot of friends of mine who've been looking at different systems for a very long time, is how to deal with that. Well, when are these processes important, for how long? I think you probably could do it with that data set.
Yeah, I think so. These data, I mean, this has mostly been under the impetus of Diana asking me to come here and talk, and me saying in a moment of weakness, oh yeah, I can do that, I'll do that. and so just in trying to assemble some of this stuff, really mostly to pay homage to all my poor summer students like Scott here and Allison in the back there, and actually Amanda Kahn, your new faculty member, she's taking my class too. But just to pay homage to all their effort in these classes, just started sort of just skimming the surface of all these data. And so in my retirement, I'm planning to sort of dig into them a whole lot more and dig out some of those kinds of things, hopefully. And then also it's just a gold mine of stuff in these photo quadrats, except I have like now 600 some odd images to analyze, looking at the cover of the benthic Cecil invert stuff.
Let's forget that it'll tell a story.
Yeah, yeah. Well, they probably will, and it'll be really interesting. But there's just been a whole lot of stuff that's happened since the times that we started doing those photographs. So it'll be interesting to do. But yeah, that's definitely. I think there's a lot of stuff that will be mined out of these data.
When we were talking during lunch, you said that the progression
No, the really interesting thing is that where the loss of the kelp canopy has stopped, it hasn't progressed further than that since last summer. And you look at the abundances of the urchins in there, and it's not quite as high as it is out toward Point Pinos. But more interesting is that the otters are rafting. They, like to wrap themselves in the kelp, like the tour boats always say. They wrap themselves in the kelp to keep warm, but they're just keeping themselves from drifting away. But they aggregate where the remaining canopy is present. And so if they are starting to eat some of the urchins, that might be one of the things slowing down the retreat of the canopy. And so at least since last summer, the position of the canopy really hasn't regressed any further back toward the harbor. And so your study site might be okay.
Great.
Allison.
So along with the nice, simple, otters eat urchins, urchins eat kelp story, there's also a more nuanced, positive feedback loops that in theory maintain barrens or kelp forests. And I'm curious what sorts of, since you have sort of the most consistent natural history knowledge of anyone probably around, especially of Hopkins and surrounding areas, what do you think happened or could have caused this change and those sort of disturbed those positive feedback loops to allow you to have some recruitment or to, make that change?
Yeah, that I'm not sure. Because the pulse, I think there's got to have been more than just 1 year, and maybe it's been like 6 or 8 consecutive years of good settlement and survival, which was at least in the times that I've been watching these places, really unusual. So that's one thing is whatever, because you guys stay in the plankton for weeks, and so the oceanographic conditions that have shifted to the point that more larvae are transported into southern Monterey Bay right now would probably bear looking at. But the other thing is just the dynamics of what goes on with the urchins. And I know studies have been done to try and look at whether they're competing for space in crevices and so forth. But one of the things I keep thinking about as one of the triggers for the change in behavior of the urchins, because most of the time, as many of you know, they're sedentary, sit in a crevice, eat drift kelp, and then when something reduces the abundance of the drift kelp, they get hungry, come out of their crevices, start grazing actively, and create the deforestation. But the other possibility, the other possible mechanism that I keep thinking about is just total number of urchins and availability of shelter sites. And even if there's abundant drift algae around but not enough crevices for them, then the ones that don't have space in the crevices just wander around, and as they're wandering around, they mutter in their urchin-like way, and they just move their little jaws, and they just start taking stuff out, maybe just because they're pissed off because they don't have a crevice to sit in. But they move around, and just that activity, moving around, starts the deforestation, even though there might be enough drift algae around. And so, again, the changing suite of potential circumstances that might cause switches in their behavior could be important elements that contribute to that. But I'm just sort of waving my hands and BSing. I have no idea, but it's a really good question. John.
So you and Chris Harold documented a big urchin recruitment on the Pinnacles. And what took them out?
Good question. We're not sure about what that was, because that started probably 1984 was when the recruitment occurred. By 85 was when we started monitoring the deforestation. And by then, there was urchins all over the place and no kelp. And so that went on till 1988, and January 1988 was a series along the whole California coast of some of the biggest storm waves that had been recorded. Those are the same ones that down in San Diego had just sort of ripped up the Point Loma kelp bed in 88. And waves were breaking over the top of the Pinnacles during that event. And so it could be that by then the urchins were hungry enough from not having much to eat and weakened enough that a good portion of them just got swept off the reef at that time, and then that was enough of a break to allow the kelp to re-recruit, because there were kelp canopies all along the near shoreline areas out there, and so there was plenty enough sources of spores for kelp to get out there and recruit as long as the grazing was reduced. And so I don't know for sure, but I always thought it was those big storms in '88, because by June of '88, when we went out there and sampled for the last time, urchin abundances were well below what they had been. But they had been declining over the years.
The drop that we had in '84, '85, that was disease. Yeah, I've been waiting for a disease to come and sweep out the things.
Well,
Disease also
could
took
still
out
happen.
Santa Cruz.
Yeah, it could still happen.
So do you think these urchins, have you looked at the gonads? Are the gonads something that the otters would bother with?
Well, again, I made my summer class do this. We sampled them last summer, which is not peak gonad development time. And interestingly, the Franciscanus at Hopkins and at Point Pinos had about 3 to 4% total mass that was gonad. So they weren't really all that different. But the Purpuratus had no gonads out at Point Pinos and decent 3%, 4% gonad development at Hopkins. And so I think the guys out at Point Pinos are starting. No. But I should go out there, more winter time, January, February, when they're sort of peak spawning, but it's windy and rough and ugly winter out there. And I don't have any students around at that time to drag out there with me. But that's what we should do is take a look at during the peak reproductive time at what they look like. But I suspect the ones out there, are not gonna be all that well developed.
The North Coast, but there's no otters, of course, but the urchin fishery for Franciscanus has collapsed because there's no gonad.
Yeah. So that's the other variable that gets into this mix is we always think of otters as just automatically eating urchins when they're there, but without really thinking about what quality of food those guys are, because it's good roughage right now, but not much else, because there's not any gonad development.
So I'm kind of depressed. I'm thinking, this is, back when I started at Santa Barbara with my PhD, the first seminar I heard was yours. I said, geez, what? I'm going to do something else. And, I appreciate the long time series thing. So what advice do you have for students at the master's or PhD level who are going to dive into kelp ecology that don't have that opportunity to use long data sets?
I think the most important thing is to concentrate on the biology. Find some of that biology that just sort of grabs you by whatever parts are most important to you, and makes you want to study that, regardless, and learn as much as you can about what we already know about how these systems work, and take it all with a grain of salt. And don't assume that what people like me who stand up in front of groups and talk about stuff really know what they're talking about. and that there's always room for learning more about how those things work. And that, again, because you read the literature and you see all these experiments have been done and they show us this and that and this and that. But a lot of those experiments, if you ran them again, probably wouldn't get the same results. And taking that to heart and realizing that automatically then tells you that there's lots of holes still in the stuff that we know, and holes that can be filled in. And a lot of this stuff that I've sort of had the privilege and opportunity to sort of observe, it's taken 40 years to do it. And so don't feel that you have to have that same level of experience of things in the short amount of time you have to do the work that you have to do in front of you. But do it with those kinds of backdrops in mind, and think of where what you find out with the resources you have and the time that you have to do it, how that's going to fit into that broader pattern of fluctuation, and not think that it's, like I said, like it's a brick in the wall that's going to sit there forever, like the Sistine Chapel or something, but that it's a set of moving parts, and you're adding one of the teeth to one of those gears that's going to turn and go somewhere else and do something else to something else, some other thing down the road that may be way different from what you can predict now. But if you don't put that little tooth on that gear, we're not going to find that out. And so do what you can as well as you can with the view of these big things that have gone on in the past. And don't be depressed about silly stories that I tell.
But I want to say one thing, and that was, thank you for saying yes.
Oh.
Because that forced you to come here,
Yeah.
and that was an amazing view of the past that not many of us had. And I've told all the students here, talk to people like you, because you're an amazing resource, and there are many other of us sitting in this room. And so I like your point about find out what other people have done, but also find your own piece in it. But talking to Jim, because he's right down the road as a resource, and Foster and Dave Shield and all the other people too, taking care of. But we wanted to say thank you. Do you want to ask? Are you guys have one more question? Yeah. This is a really special seminar from Jim.
Excellent.
Okay, one more.
Just really quickly, I've heard suggestions that perhaps it would be a good idea to go out and kind of get rid of a bunch of those purple urchins. What do you think about that? Or anybody, the other Kelp Force folks in the room?
I am not an advocate of us screwing with nature. It's because before we were here, probably these kinds of events happened all the time. And, if you can think of the ecosystem as an entity, it knows how to deal with this sort of stuff. It might take a little longer than we would want, and it might disrupt what we feel is the way things should be, but maybe our view is not quite of what they should be isn't quite right, and this fluctuation is what it should be. And so I think to me it's more interesting to accept that it's happening, and that at least in this instance, at least as far as I know, this is a natural event. This isn't something caused by us directly, at least. And so let's sit back and watch it, because it opens our eyes to a lot of stuff, like the nuances of that sea otter, sea urchin, kelp interaction that we would have missed if the urchins hadn't showed up, because we would have just kept thinking, oh, the otters are here, they're eating all the urchins and they're protecting the kelp beds. But because this thing did happen, this is a chance to learn more about it. And I would be real hesitant, since I don't think we were wise enough to make the decisions on how to screw around with nature that way. That it would actually, we'd even be able to do anything, remove enough urchins to actually make a dent. Because Bob Stenick tried to do this in Maine, and they removed, millions of urchins, and it didn't change the ecosystem at all, because more of them recruited back in and filled in right after they removed them. So I would be much more an advocate just to watch and see, because that's the interesting part.